In the previous video, we have understood or we have discussed that it's the STK11 cell lines from the knockout library of murine genes. It is 915 murine genes, and each gene has 5 guide RNAs. So from that knockout libraries, the based on their bioinformatics analysis, they are able to find out that STK11 is the one that leads to the most migration in the pancreatic cancer cells, that is PANC1 cells. So they decided to check whether STK11 gives what kind of migrations STK11 gives compared to the wild type cells. So they developed a single knockout cells from the pancreatic cancer cells, and also this TB32047 cell lines. And in these cell lines, then they compare the migration based on the scratch wound assay on InQ sites. So the first figure is to show that the knockouts are bona fide. So what they did is they checked their expression of this STK11 knockout. They did the around 5 different knockouts in single cell lines. Here they have presented data for 2 knockout cell lines. First one is the wild type, where they can see the relative fold expressions, and which is been considered as 1. And in the case of this NC, that is control cell lines, they do not see the significant difference of the relative expression of this STK11. However, for the knockout cell lines, that is STK11 KO 1.5 and STK11 KO 2.2, the relative expressions are statistically lower compared to the control cases. And similar observations are found for the PANC1 cell lines, just to validate that they have the bona fide STK11 knockout cells. And after that, they also did the Western blot to confirm this knockout of STK11 using completely different approach. That is what usually done in the biological experiments. That is, in this case, they do not see the signals for the STK11, neither in the TV32047 cell line, neither in the PANC1 cell lines. And after that, they did the Sanger result sequencing also. Those results are presented in the supplemental figures. Thereafter, after validating that they have the bona fide knockout cell lines, they did the scratch wound assay on both the cell lines, that is TB32047 and PANC1 cell lines. And what they have find out that after 24 hours, the motility of this knockout STK11 knockouts are higher compared to the wild type, and the quantification of this panel E is in the panel G, where we can see that after 24 hours uh, this STK knockout 1.5 and STK knockout 2.2 STK knockout 2.2 are the percentage of yield between of this STK knockout 1.1 and STK knockout 2.2 is very high, or is higher than the percentage yield that they have find out in the control case and in the NC 1.4. Around we can say that 65 to 90% is shown in the STK11 knockout cell lines, and around 30% is shown in the control cell lines, of TB32047. And this indicates that the how this substantiate their previous finding that STK11 knockout has higher migrations compared to the wild type TB32047. And similar experiment they did for the pancreatic PANC1 cell lines also. And here also they find those similar results, although the difference is lower than the TB32047. So what they have find out after 24 hours, that in the case of this control, the percentage yield is around 55%, whereas in the case of knockout cell lines, the percentage yield is around 75 to 85%. that can be seen in the panel F, and its quantifications are shown in the panel H. So from their migration, scratch wound InQ site migration assay, they are able to substantiate their previous finding that STK11 genes are important. STK11 genes are STK11 knockout cell lines are have STK11 knockout cell lines have more migration properties, and they have more mesochemical properties. Mesochemical properties are not shown here directly, but the migration properties are being shown here using InQ-site scratch wound assay, that the STK11 knockout cell pancreatic cells has more migrations properties compared to the wild type their respective cell lines. So I hope you understood how they have shown their how they have substantiated their finding from the previous experiment that STK11 has the more migration properties using scratch wound InQ-site assay. If you need to understand more about their experiments in detail and their supplemental figures, if you also want to refer to the supplemental figures, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.